Okay, so I'm Jenna Ridlin, and in my bioinformatics class this semester, we are partnering with Hudson Alpha Code Project. And actually, our Hudson Alpha counterpart and founder of the Code Project is here in our audience right now, Michelle Morris. And so I'm very, very excited to present one of the projects from our class. This is Joanna Lapoint, and she is going to be talking about a variant of unknown significance in a gene that we're studying collectively, which is CTLA4. And I'll have you take it from here.
All right. Hi, I'm Joanna Lapoint. My presentation is titled Examining the Pathogenicity of VUS R75Q in the human gene CTLA4. All right, so CTLA4 is located at 2q33.2, that is its chromosomal location, chromosome 2, band 33. CTLA4 codes for cytotoxic T lymphocyte-associated protein 4, which functions as an immune checkpoint and regulates an immune response by transmitting inhibitory signals to T cells. It is a member of the immunoglobulin superfamily, and it is a homodimer that is connected by a disulfide bond. Diseases associated with mutations in CTLA4 are type 1 diabetes, Graves' disease, Hashimoto thyroiditis, celiac disease, systemic lupus erythematosus, thyroid-associated orbitopathy, and various other autoimmune disorders. So the variant R75Q is located on exon 2 at C.224, guanine over adenine, P.arginine 75, glutamine. I selected this variant due to its uncertain clinical significance and close proximity to a known pathogenic variant at location 75 as well. The CAD score here, so CAD is combined annotation-dependent depletion. I have both the raw and scaled score, which both indicate possible damaging effect. CAD predicts deleteriousness of single nucleotide variants. I'm a little nervous, sorry y'all. Okay, so the amino acid and structural comparison of VUS R75Q. In the wild-type protein, arginine is located in the 75th position in the polypeptide chain in both molecules of the homodimer complex. So arginine is a positively charged polar amino acid. It will most likely be found in a position on the surface of a protein. It can form ionic bonds with negatively charged or acidic amino acid groups. Due to that positively charged NH group here off of that carbon. Ionic bonds can be almost as strong as a covalent bond. And its location in the outer beta sheet within the of each polypeptide indicate that it is important to the protein's tertiary structure. Glutamine, it is a polar amino acid with a shorter side chain. The oxygen in this side chain over here allows for weak hydrogen bonding. So this variant results from a swap from guanine to adenine in the second nucleotide position of the codon CGG to CAG. If the protein structure requires the electrostatic interactions from arginine, then the mutation to glutamine can alter the structure and or affect its stability. The cellular expression of CTLA4, it's found on the external membrane of cells. Right, let's see if I can get the point. Here we go. It is a receptor protein that binds ligands such as CD80 and CD86, which are found within the membrane of antigen-presenting cells. This protein has 3 domains, an extracellular V domain, a transmembrane domain, and a cytoplasmic tail. And it is located in, the variant is located in the extracellular domain of the protein. This is a USARA-scene. USARA stands for yet another scientific artificial reality application. It is a bioinformatics software that creates 3D models and variant simulations. This is my wild type with the arginine and its longer side chain here on both molecule A and B. And this is my wild type, my variant here with the shorter side chain, right there in the folding region of the beta sheets. This is my polyphen 2 prediction. So let's see. Polyphen-2 is a tool for the prediction of potential impact of single nucleotide polymorphisms on the structure and function of proteins. I have an interesting thing here where my HUM-DIV score indicates that this is a probably damaging variant, where my HUM-bar diverges and says benign. Here is a multiple sequence alignment generated by polyphen-2. So the difference between HUM-DIV and HUM-bar, it may be, HUM-DIV is compiled from all damaging alleles with closely related mammalian homologs assumed to be non-damaging. That one, hang on, let me start that over, I'm sorry y'all. The HUM-bar may be benign due to both arginine and glutamine being hydrophilic, and the reference proteins used by this program may not require ionic bonds. HUM-DIV may take into account the electrostatic interactions between amino acids and reference proteins. However, we need to further look into the discrepancy. The GERP and ARVIS score. GERP is a genomic evolutionary rate profiling program. It measures the evolutionary conservation of a genomic region. This score indicates at 4.2 that it has a relatively high level of conservation. ARVIS is the residual variation intolerance score. It indicates that the gene's intolerance to variation is moderately intolerant at negative 0.01. This is my Sift prediction. Sift predicts SNV effects on protein structure and function based on the sequence homology and physical properties of amino acids. It is useful for accounting for the positive and neutral charge difference in arginine and glutamine variation. This prediction is damaging. This is a multiple sequence alignment from Benchling. Benchling is a bioinformatics program that allows the import and analysis of full gene codes and cross-species comparisons at both the nucleotide and amino acid levels. So this MSA table includes 4 mammals and 1 bird and 1 fish. Unfortunately, no reptile or invertebrate cDNA files could be found for import, though Ensemble does show that there are orthologous genes within those. So more research will be needed to determine the conservation between reptiles and invertebrates. However, it does show that it is highly conserved across mammals, and not fish, but the bird, G. gallus, is a red jungle fowl, and it maintains the arginine at the amino acid position, which is this table 2 here. It maintains the arginine, but its codon is slightly different at CGC. The only change here is in the wobble nucleotide position, which is usually not, does not usually affect the amino acid outcome. So the level of conservation does indicate that it would be a deleterious change. So in conclusion, it is highly conserved among mammals. The VUS is conserved across at least 1 species of bird. The divergent polyphen-2 results show that more study is needed to determine whether or not the VUS is truly damaging. However, all the other data gathered and presented here does indicate that it is likely so. Further study is needed in both model organisms and patients with known variant to officially describe it as pathogenic or not. I'd like to thank the JSU College of Math and Sciences, the Department of Biology, Code Hudson Alpha, and Dr. Jenna L. Ridlin. And a special thank you to Dr. Sauter for his thorough instructions in cell biology last semester. I didn't have to look up any of that. And also to Dr. Turner, who can't be here, for her thorough instruction in genetics last semester.
Questions for our speaker?
I'm kind of curious, can you show those 2 images of the tertiary structure thing with the arginine and the glutamine? Okay, let's see. All right, so that's in the 3rd one. Let's go to the oh, okay, so really didn't shift the structure around that much. All right, I'm just curious.
Yeah, I actually have one I didn't take a screenshot of, but I have one where both of these are overlapping and it mildly changes the shape right there.
For a minute, I thought like really shifted 2 or 3 chains out pretty dramatically, but all right, it hasn't changed. Okay.
Any other questions? Let's thank our speaker.